Hey everyone, and welcome to today's Explainer. I am so thrilled to have you here with us. Today, we're taking a journey into the incredibly powerful microscopic forces of the posterior pituitary gland. You know, it's this tiny little structure sitting quietly right at the base of your brain, but as we're going to see, it plays an absolutely massive role in both our physical survival and our deepest social connections. We're actually calling this one the heart and the plumbing because I think it perfectly captures the fascinating, kind of split personality nature of the hormones we're about to uncover. Okay, let's dive right into this. What exactly creates the unbreakable bond between a mother and her child? It's so easy to think of love and connection purely as abstract emotions, right? Or just psychological phenomena. But the reality is that this profound emotional connection, that fierce, protective, nurturing instinct, is actually heavily driven by chemistry. It's literally a biosocial masterpiece engineered right inside the human brain. Enter the brilliant molecular architect of that bond, oxytocin. People often affectionately call it the bonding hormone, and honestly, it is remarkably small. Chemically speaking, it's just a 9-amino acid peptide, a nonapeptide, that features a very distinct disulfide bridge holding its structure together. It's actually synthesized originally as a pre-pro hormone up in the hypothalamus, and then it travels via specific carrier proteins down the axons just to hang out and be stored in the posterior pituitary. But seriously, do not let its tiny size fool you. This simple little chemical structure is the biological foundation for how we and other mammals modulate maternal nurturing and deeply complex biosocial behavior. All right, moving to section 1, oxytocin and maternal bonding. Let's explore exactly how this incredible molecule goes to work in what we'll call the heart of the posterior pituitary system, where strict biology creates the warm foundation for human connection and life itself. To really wrap our heads around oxytocin's power, we have to look at its role in parturition, which is just the medical term for childbirth. It operates on this beautifully orchestrated classic positive feedback loop. So, step 1. As labor begins, initial uterine contractions physically stimulate stretch receptors located in the uterus and vaginal mucosa. Step 2, this physical stretch sends a desperate signal straight to the posterior pituitary to release a massive surge of oxytocin. Step 3, that newly released oxytocin acts directly on the estrogen-primed smooth muscle of the uterus, which causes even stronger, further uterine contractions. This creates an amplifying loop: more stretch, more oxytocin, more contractions, until finally, step 4, childbirth is successfully achieved. This biological mechanism is so reliable and so potent that synthetic oxytocin is actually used therapeutically every single day as a standard medical agent to induce labor. Now, what's really interesting here is how we can visually trace the dual pathways of maternal bonding and milk letdown. If you follow the arrows, you'll see a neural loop and an endocrine loop working together in total harmony. When a newborn, like the calf you see here, stimulates the mammary gland by suckling, that physical sensation travels all the way up the spinal cord via a neural pathway straight into the mother's hypothalamus. The hypothalamus immediately responds by triggering the posterior pituitary to dump its stored oxytocin right into the bloodstream. And that right there is our endocrine loop. This oxytocin then travels back down to the breast, stimulating the contraction of specialized myoepithelial cells, literally squeezing the milk out of the alveoli for the infant. So the physical act of feeding isn't just delivering vital nutrition. It is literally flooding the mother's biological system with the exact hormone that deepens her nurturing maternal bond. It's brilliant. But here is a crazy twist. Did you know that emotional stress can actively inhibit lactation? This highly sensitive oxytocin system relies on a delicate intracellular mechanism called the calcium PIP2 pathway. When a mother experiences high emotional stress, her brain can actually suppress this exact pathway, meaning the milk is fully present in the mammary glands, but the smooth muscle simply won't contract to release it. It's a really powerful, tangible reminder of how deeply our psychological state is intertwined with our physical biosocial systems. Which brings us to Section 2, ADH and water balance. Now that we've thoroughly explored the heart of the matter and the incredible social power of oxytocin, we need to shift gears and examine the body's strict internal plumbing. To bridge these two physiological worlds, let's look at this side-by-side comparison. On one side, we have oxytocin, our bonding hormone, stimulated by suckling and stretch receptors to create maternal connection and smooth muscle contraction. On the exact opposite side of the functional spectrum, we have its chemical sibling, arginine vasopressin, also widely known as ADH or antidiuretic hormone. They are remarkably similar in their base structure. They're actually both nonapeptides featuring that same disulfide bridge, yet their jobs honestly couldn't be more different. ADH is purely about internal survival mechanics. Its main stimulus is an increase in plasma osmolarity, which basically means when your blood gets too concentrated or salty, or when your arterial blood pressure drops. Its absolute crucial function: strict water retention and vasoconstriction. So the crucial point here is exactly how ADH forces the body to conserve water and raise blood pressure. It accomplishes this massive task through some very specific receptor pathways. The V1A receptors use a GQ protein mechanism to essentially trigger widespread vasoconstriction, clump platelets together, and even cause myocardial hypertrophy in the heart muscle. V1B receptors, on the other hand, mediate the secretion of ACTH to help manage stress. But the real magic of fluid balance, that happens at the V2 receptors, which operate via adenylyl cyclase. These guys specifically target the renal collecting ducts in your kidneys, stimulating the insertion of specialized water channels called aquaporin 2. These channels rapidly increase water reabsorption, rescuing free water from becoming urine and pulling it straight back into your bloodstream to dilute your plasma. And now let's hit section 3, when hormone balance fails. We've seen how precise and powerful ADH is when it's functioning normally, but this brilliantly illustrates the extreme, honestly life-threatening consequences of an ADH system gone totally wrong. On the right side, we have diabetes insipidus, or what we can call being dry inside. In this state, the body has lost its antidiuretic hormone function, leading to massive unchecked fluid loss, extreme dehydration, and a really dangerous concentration of blood sodium, known as hypernatremia. Over on the left side, we have the exact opposite extreme: SIADH, or Syndrome of Inappropriate Antidiuretic Hormone Secretion. Here, the patient is essentially soaked inside. The body autonomously produces way too much ADH even when there's no known stimuli for it, locking water inside the body, diluting the blood to cause hyponatremia, and producing very sticky, highly thick urine. So when clinicians are trying to diagnose these polyuric or high urine output states, we have to look closely at where the mechanical breakdown is actually occurring. With hypothalamic diabetes insipidus, or central DI, the failure is right at the top. The pituitary gland completely fails to secrete AVP, and this is often due to head trauma, neurosurgery, or tumors. Then you have nephrogenic diabetes insipidus, where the pituitary is firing perfectly fine, but the kidneys just ignore the signal, usually due to a mutation in those V2 receptors we talked about, or certain drugs like lithium. And then there's psychogenic polydipsia, where excessive chronic water intake naturally suppresses AVP secretion. To give you an idea of how severe this gets, with a complete AVP deficiency, the failure to reabsorb solute-free water means a patient's urine output can approach an astonishing 1 liter per hour. Can you imagine? Let's move on and see how this builds into a really vital diagnostic matrix. Clinicians use this exact setup to decode the problem by comparing blood and urine markers. In SIADH, because the body is inappropriately retaining way too much water, the serum sodium and serum osmolality are heavily diluted, so those numbers go down. But the urine is highly concentrated, so urine osmolality goes up. In normal severe dehydration, everything is concentrated, so all 3 of those markers go up. But in diabetes insipidus, the blood is super concentrated because water is being rapidly lost, so serum sodium and osmolality go up while the urine is completely diluted, bringing urine osmolality crashing down. This matrix is the ultimate key to understanding hemodilution versus hypernatremia. Now, the human body is incredibly resilient and actually has a built-in failsafe for the dangerous fluid overload we just saw in SIADH. When all that excess water dramatically expands the blood volume, it physically stretches the heart. The cardiac atrium responds to this stretch by releasing atrial natriuretic peptide, or ANP. This peptide travels straight to the kidneys and forces them to excrete sodium, a process called natriuresis. Water naturally follows the sodium right out of the body, successfully reducing the blood volume. I love this because it proves just how deeply and beautifully interconnected our cardiovascular and endocrine systems truly are. It really all comes down to this. These 2 tiny 9 amino acid chains dictate both our survival as individuals through water balance and our survival as a species through maternal bonding. That is the core takeaway of our explainer today. We've traced the incredible outsized impact of these microscopic posterior pituitary hormones, from the rigid plumbing of our kidneys keeping us hydrated, all the way to the deeply emotional heart of a mother's embrace. Biology is what binds it all together. So, I want to leave you with this final provocative thought. If a microscopic chain of just 9 amino acids can build a mother's unbreakable bond, induce childbirth, and completely dictate a human body's hydration, what other invisible chemical forces are quietly authoring your daily life right now? The microscopic world inside of us is infinitely fascinating. Keep exploring, keep questioning, and I absolutely look forward to guiding you through our next explainer.